presenting. So I just will say thanks to my collaborators, and also I'd like to say thanks to Ming Zhang from NIMH, who was generous enough to provide us with support to make this possible. We very definitely hope to be giving additional webinars in the future. So if you enjoyed yourself here, be sure to tell your colleagues about what we're doing. I don't think that there are other announcements except to say that we're recording each of the sessions, and we have checked with each of the presenters to find out if that would be okay. And hopefully no one has any objections. What we'll be doing is posting videos of the individual presentations once we get them uploaded to the right platform. Let me ask my buddies here who helped put this together, Michelle or Anoop, are there any other announcements?
I don't think so.
I just put the link for the agenda in the chat if somebody wants to look at it, just to refresh their memory.
Okay, thanks very much. So first, we're going to start with a presentation from Mike Horowitz, and he will sort of walk us through the kind of the data ecosystem that we have for our overall infrastructure, and then I'll have some remarks after that. So go ahead, Mike.
Okay, thanks, Owen. Let me share my screen here. Okay, can people see this PowerPoint?
Yep.
Yeah. So these are I'm going to share some slides here just on the BICCN in general, Brain Initiative Cell Census Network, and some of the related kind of data points concerning with it. It's just meant to frame kind of some of the NEMO kind of work and activities. I won't going to talk about NEMO at all. Obviously, this is going to be, I'm going to hand that over to the experts momentarily, which will tell us about the whole workshop and the day. Basically, NEMO is part of this big ecosystem of the Brain Initiative. In addition to other awards, they serve the Brain Initiative in a big way, the BICCN. This is a project whose goal is to generate a really comprehensive reference atlas, cell type level reference atlases of the mouse, non-human primate, human ultimately, and to understand the molecular and atomic and functional aspects of these. The project at this point is expansive. This is not an ego. I didn't make this slide. I'm borrowing this slide, but it shows the kind of the landscape now of there are what started with approximately 6 awards a few years ago is now substantially up into the 20s, and it contains lots of things from all kinds of molecular profiling techniques, epigenetic techniques, anatomic and tracing techniques, and electrophysiology as well. Importantly, all this data and all these PIs are the data is sort of served and stored in these archives, of which NEMO forms a critical one. This is what we'll be learning about here today. NEMO houses, of course, all the single-cell transcriptomic data, the epigenomic data, and which is the largest part of the project, not necessarily in terms of data scale, because imagery is very, very large, but in terms of certainly in terms of cell numbers. The technology focus here is the idea was to bring to bear all these modern aspects of single cell profiling in a collaborative way from multiple labs, multiple PIs done to basically kind of coordinate this kind of large scale profiling exercise. And the consortium itself has been, I think all of us would probably agree, a terrific success. in its generation of molecular signatures, anatomy, and bringing kind of all these techniques to bear. Each people, different groups, different PIs doing different approaches, but the discussions and the synergy to them have been really remarkable. This has is led to a paper, a collection of papers, in addition to a main manuscript summarizing the overarching findings. There are approximately 8 companion papers that go along with these, which are done for each of the various modalities and techniques. This is just showing here a piece of figure 1 from the platform paper, which is now in review at Nature. Well, we've received the reviews. They were very favorable, and we're working to kind of put together some of the final touches to it. Importantly, that what does this ecosystem look like for all this data, and how does sort of NEMO and the archives fit into this? Is that we want to provide access to this data for users to basically follow up with, to continue studying of this, to make the data have a legacy value. Over here you can see this is the Neuroscience Multi-omic Archive, also the Brain Image Library, and a more recently added archive, the Dandy Archive here. These are sort of centrally kind of hooked into this brain cell data center, which we kind of maintain here at the Allen Institute. And this is now we have kind of some basic kind of sort of display and summary methods for some of this data. This links downstream into various analysis tools, for example, PSNE and differential expression kind of mapping, as well as importantly, the tools from NEMO Analytics here, which I'd like to mention, and I know that we'll be hearing about that in the course of the workshop. So this provides ways of analyzing the single cell data, and you can learn about that a little bit later in some of these sessions. There's new efforts going on. This was just the first effort of this. We picked this first project, which was a deep profiling kind of the primary motor cortex, and it was started in the mouse and then extended to do a cross-species mapping, and part of the flagship contains a real cross-species analysis of this corresponding area in human. Several projects here that I'm just mentioning here of these new efforts that are sort of taking it to the next step. This includes extending a lot of the profiling that was done to whole brain in the mouse, focusing in a much more comprehensive way on molecularly annotated wiring diagrams, a deep study of neural regulation elements and how they connect with the cell type clusters, and a first take at a proteomic atlas. So the BICCN, this BCDC serves as a big kind of coordinating engine for the BICCN. We do this with various working groups that are very active. Many of the people who actually put this workshop together are very active in the infrastructure working group, which is perhaps one of the most important ones as far as orchestrating and organizing data and making sure that it flows between the various groups. And there are many others. And we also have, we aspire to and have connections with other programs. I certainly would call out Dr. Yang Yao from NIH, who has been a champion of this and is the main program officer for the BICCN, and does a lot to keep us with the right focus on the big picture. If you go to BICCN.org, you can see all these different projects listed. There's sort of pages for each of the PIs and each of their projects. Now we have a new kind of data kind of access sort of template here, which is sort of a cell survey. That basically each of the projects gets a description and a direct link. For example, you can link directly into the NEMO archive to, for example, get at the data which was generated for these various experiments. Finally, there's lots of resources available to learn more. You can go to the BICC website. I know we'll be hearing a really nice tutorial from Tim on the Broterra. There's the NEMO archive, NEMO analytics, the brain image library, and the Dandy archive, as well as neuromorphology.org. All the platform papers I mentioned are accessed. The platform paper and companion papers are available on the bioarchive at these links here. So I think without further ado, I really want to hand it off back to Owen and let them kick off this really nice day. So thank you.
Thanks, Mike. I will grab the screen as soon as you stop sharing.
Okay, let's see. We're going to find my stop sharing button here. For some reason, it seems to have gone away. I'll just exit and come back.
Okay.
Okay, there we go. Yeah.
All right. Is that visible?
Yep.
All right. So before we get started on talking about our individual resources, I just wanted to zoom back a little bit further. Mike gave a great introduction, but I just sort of want to talk a bit about what NIH is also trying to achieve across all of the institutes with respect to hosting data and making it more accessible to people. And what you'll see is I'm going to talk probably more about the challenges than the good news stories, but there are good news stories. So everybody understands this, that the biomedical research landscape has gotten larger and larger. I think the Broad right now is averaging one human genome every 5 minutes right now. And we're certainly generating data from millions of cells, and the data generation is being distributed from thousands of centers now, which is a good thing. And we also have a lot of cloud-based systems for storing data, as well as abundant resources. I'm not sure where I got this number of 180 million. I think it was an old number, but obviously the resources behind data generation is huge. So when we start thinking about the world that we'd like to live in, there are lots of different data coordinating centers that are out there. These represent data coordinating centers from Common Fund, but we have assets that I could just generically, a term I could use to generically refer to lots of different types of files, from RNA-seq data or variants or patient phenotypes. And they're hosted at many different sites at this point, and that's never going to change. We're not going to go back to one big, huge repository. And what we'd like to do is increase our ability to discover that data and make use of it. And what I'd especially be interested in doing is enabling the clinical researcher. I'm part of a project associated with Common Fund and looking at all the assets that are available from their data coordinating centers. And that's what this is meant to show, which is this is essentially a grid from a lot of the different data coordinating centers and the different modalities of data. And you can see that there are many data types that are found in common across these DCCs. And they also exist at lots of different levels of resolution, which is exactly what Mike just said. And the important part of this is that there may be cases where you can find the same type of asset across data coordinating centers, but of course, there's also lots of complementary assets that we'd like to be able to access. And when we do that, we start to talk about and use the term an ecosystem. And one way that you could sort of think of an ecosystem is exemplified by if you go to Amazon and you want to look at purchase lots of commodities there. The idea is that there's lots and lots of different sites that are producing things like LCD screens. And we don't actually ship those LCD screens to one common warehouse. What we do is we ship metadata associated with those LCD screens to a common catalog, and then you can search on that. And one way that you could approach creating an ecosystem is to sort of do the same thing. And this is something that I frequently refer to as federation, where instead of shipping the files themselves to a central repository, we could just make a relatively straightforward way of describing the metadata associated with those files and put them into some type of catalog, and then create an interface that's not unlike the one that you could find in Amazon to allow people to do different types of review of data or to capture large data sets and then pass them on to analysis platforms to be able to make use of that information. So this is my concept, or some people's concept, of what federation is, where you're not moving the files themselves, you're just moving data associated with describing those files to a central catalog. Now, the reason that this is hard, one way to describe this, the reason that people haven't gotten further with being able to do this is that it's essentially kind of a game theory type of problem. It has to do really with fairness and trust, which is you can have lots of individual sites that would like to work with each other, but TopMed, for example, that's been around for a long time, and Kids First, which has generated variants associated with lots of genetic diseases, or GTEx, is unable to necessarily get the other DCC to decide to use the same standards for describing their metadata. And so what you have to do is kind of create a world where the stakeholders have a vested interest. And Mike talked about our ecosystem, and we do have a vested interest in providing a really good quality product to the consumer, but it takes a long time to really build up consensus to be describing this data. And that's what we're in the middle of doing right now. Standards are definitely dependent on community engagement. We have to be working with people that are on this call. We have to be working with different data coordinating centers. And it takes a long time. It involves setting up working groups, and it's very collaborative and very iterative. It's not something that you can really stand up overnight. So if I were to describe to you in really quick terms, what is a data ecosystem? We're going to show you an example of essentially a nascent data ecosystem, but there's progress that we could be making. It requires federation. So the idea here is there are examples of data federation standards. A really big one is schema.org. It's not necessarily genomic. Another one that you may have heard of is GA4GH, which is a project to essentially federate across a very large number of resources that have generated specifically human variant data. Another example of data federation could be the UCSC browser that has something called track hubs, that if you've got a reference genome, people can agree on essentially giving genome annotation along that reference genome. Another fantastic data federation project is the FHIR system, which is essentially a data model to allow hospitals and other patient care services to be sharing data. And I'm involved with a project for something called the cross-cut metadata model, which is coming out of Common Fund. And as I've mentioned, we as a group, BCDC and the other data archives, are trying to make a federated system as well. And you're going to see lots of examples of that really working in a pretty reasonable way. Another huge problem that we're always confronted with respect to data ecosystems is that they require having some amount of regularized data. And if I use the term regularized data, it's shown pretty well. Like if you go to eBay and you just take a look at their different categories, suppose I'm interested in buying a hammer, I would probably pick hardware as one of their categories, but it actually turns out that you could find a hammer in lots of different categories. And that in that sense, there's like it's very poorly regularized in terms of the way that you might be able to, you could find it in antiques, or you could probably find it in sporting goods, and you could find it in toys and hobbies. And it's made worse by the fact that if you go to another commercial site, they have a completely different set of categories that they would use to be describing data. And it's also kind of a mess when you want to go find some type of commercial product. We're faced with that in the health domain, where ICD-9 happens to be one of the ways that lots and lots of different hospitals describe if a patient comes in and presents with some type of thing that you could possibly call a disease, but a broken leg is not a disease, and broken leg is described in ICD-9. And it's also just kind of a mess where lots of different hospitals may use this category system or use different types of categorizations for sharing information. What I mean really by regularized data is exemplified by some work that's being done out of disease ontology, where they've created this large controlled vocabulary for terms that would describe things like disease that make sense to a biologist, but also they've cross-linked a lot of the existing terminologies like ICD-9, ICD-10, UMLS into this disease ontology. And so if you use the disease ontology, there's a way of mapping from one system to another. And this is what I mean by regularized data. So what's nice about regularized data is if you get your act together, if we get our act together well and start describing cell types, which is a project that the people at the Allen Institute are busy doing, then we would have a fighting chance of asking questions like, give me all the RNA-seq files or all the single cell data associated with a specific cell type, which is really, it's going to be a while before we tackle this problem, but this is part of what's needed for a really healthy, robust ecosystem. So data ecosystems also require analysis platforms, and we're going to spend the rest of the day convincing you that this is a thing. The idea is that there are platforms out there that don't just give you access to data, but they essentially supply a number of layers of very important sort of a kind of a software stack that enables you to not just get data, but handle things like authentication, wade through the metadata that's associated with them, use something like a global unique identifier, like a DOI, to be able to retrieve data, and then perform some operations on them. And this is the problem, is that we kind of live in a space right now where there are, if I use the term stack to describe this multi-layered system of making data available, there are very definitely NIH projects that are associated with this, GTEx or TopMed. Lots of these people have these operations have been implemented on a stack, but they don't necessarily very effectively talk to each other. And that's the problem that we're kind of faced with right now in the NIH landscape. And then the last thing that I'll say is that an ecosystem is highly dependent on training. I really don't care about standing up any of these things unless we're enabling clinical researchers or other researchers that are trying to publish or to execute on grants. I don't want to do any of this unless we're really building up the capability for you. This is the most important end product of everything that we do. So I just wanted to say that's why we have stood up this project. That's why we have stood up this webinar, and we hope to be having many more. So with that, I'll just end and say welcome and ask people if they have any questions, or we could move forward to the rest of the webinar. Pause for questions. Any comments from people? Okay, in that case, that was your brief primer on what an ecosystem is, and we'll spend the rest of the day going through.
Actually, can I ask one question, Owen? Just to perhaps as you know, the kind of the NEMO lead, can you just give your perspective on your vision of NEMO's role, particularly with the BICCN and thing? I think it'd be nice to hear. I mean, it's sort of, we can deduce it, but it'd be nice to hear it from you, I think.
Yeah. So thanks, Mike. That a wonderful softball. Mike and I have been working on strategic plans for the next year. For the last month, I think. So he knows the answer to this question, but I am a big believer that there's very straightforward ways for us to engage in federation. And one of the most important elements of that federation is first describing the assets. So just creating inventories of the individual files that all of the data producers have created, but also describing collections of data. So we don't really care about any one RNA single cell seq file these days. We care about what it was grouped together. And then we want to make robust metadata to be able to describe those files so it's useful to people who are on this phone call. And if we do that and sort of create kind of a central way where that would be, it'd be possible to interrogate that, like if a user could come to the BCDC and discover all of those data sets, which is obviously, that's the role of BCDC, but there are lesser or greater ways that we could really robustly be describing those files. And then if you take an investigator like Ed Lean or Joe Eckert, that they would be able to describe collections and start to share those collections for analysis and for publication with each other in a nice, robust way. That, I think, will get us out of the sort of morass that we're in, where there are many, many assets and they're still kind of hard to find. We've made a lot of progress with that, but I just think that we could go much further. So thanks, Mike. I appreciate that. So I think we'll just pass the baton onto the group because I can imagine that people are anxious to maybe start doing some work. So Michelle, who's next?
Yeah, so I'm going to first stop the recording for this session. So then we will